Bismillahirrahmanirrahim. Today the topic of our discussion is gluconeogenesis. Gluco means glucose, neo means new, and genesis is derived from generation. So the generation of new glucose molecules is known as gluconeogenesis, but this formation is not from carbohydrates but from non-carbohydrate, means non-sugary sources are responsible for the formation of the glucose. So the formation of the glucose from non-carbohydrates precursors is known as gluconeogenesis. The most important precursors are lactate, glycerol, glucogenic amino acids, and propionic CoA. First of all, we will see how pyruvate is converted into the glucose molecule, then how glycerol is converted, how amino acids are converted, how propionic CoA is converted. So we will see one by one that how these non carbohydrate sources are converted into the glucose molecules. It is not the complete reversal of glycolysis, but there are 4 important enzymes that reverse the steps that are actually irreversible. As we know that in glycolysis that we have discussed earlier, in glycolysis there are 3 steps that are irreversible, that are cannot be reversed, that are irreversible, and that are in the single direction. Those reactions are reversed by some enzymes, and then the process is reversed and the glucose molecules are synthesized. 6 ATPs are utilized per molecule of the glucose synthesized. This process is a source of glucose in starvation. When we do not eat food for 16 to 8 hours, then our body have amino acids and propionyl CoA and the other precursors of body other than the carbohydrate sources started to degrade and form the glucose molecules. And we know that glucose is the most important molecule in our body for providing energy to our body, and especially our brain and red blood cells require glucose for their proper functioning and for their proper health. 4 important sources of gluconeogenesis, as I have told you earlier: lactate, glycerol, propionyl CoA, glucogenic amino acid. What is the site? Number 1, liver. Number 2, renal cortex. So there are 2 main sites where gluconeogenesis occurs, but first of all see that in liver and renal cortex. What is renal cortex? Renal cortex is the part of kidney. In these 2 organs, the cells have mitochondria. We know every cell have mitochondria and every cell have cytoplasm. So in the cytosol, that is a soluble part of the cytoplasm, cytosol, and in the mitochondria of the liver and the kidney, in kidney the main part is renal cortex. So in kidneys, renal cortex, and in the liver, in cytosol, their cells have cytosol. In their cells, in cytosol and mitochondria, this reaction takes place. That means our body synthesizes the glucose from non-glucose sources into the non-carbohydrate sources into the liver and in our kidney. So what are the steps? We will discuss one by one. What are the steps for the synthesis? Number 1 is the pyruvate. Pyruvate is converted to oxaloacetate. We can see it step by step that pyruvate is converted into the oxaloacetate, and oxaloacetate is converted into the phosphoenol pyruvate. And we know that in glycolysis, phosphoenol pyruvate is converted into the pyruvate, and this step, that is the 3rd step, which is irreversible. There are 2 other steps which are irreversible, and the last 3rd step, that is the irreversible, that is this step of the glycolysis, when phosphoenol is converted, pyruvate is converted into the pyruvate. So the phosphoenol pyruvate is formed from this process. So we can see that from pyruvate oxaloacetate is formed, from oxaloacetate phosphoenol pyruvate is formed. For the conversion of pyruvate to oxaloacetate, pyruvate carboxylase enzyme is required, and one thing is very important here. whenever carboxylases are used, then we require A, B, C. What is ABC? A for ATP, B for biotin, and C for carbon dioxide. So whenever carboxylase enzymes are used, we require A, B, C, that is the ATP, biotin and carbon dioxide. Next, we can see that when oxaloacetate is converted into the phosphoenol pyruvate, then we have an enzyme, phosphoenol pyruvate carboxykinase enzyme, that is responsible for the removal of carbon dioxide here, and a molecule of GTP is used. So in this process, pyruvate is converted into the phosphoenol pyruvate, and when we have when we are able to form the phosphoenol pyruvate, then the next steps of glycolysis are reversed. As we can see, these are some steps of glycolysis. I can show you here. These are the some steps of glycolysis. You can see that here there are some steps of the glycolysis of phosphoenol pyruvate to the 2 BPG, 3 BPG, and up to so on up to the glucose. The whole process is reversed. So we can see one by one that how phosphoenol pyruvate is converted to glucose from the pyruvate. So now we can discuss that how lactate is converted into the pyruvate. Here is the glycerol, how it is converted into the pyruvate, and we can see how propionyl CoA is converted into the pyruvate, and how lactate is the converted into the pyruvate. So one thing is very important, when you will see I have added here the lecture. So after the lecture slide, you can see here is the glycerol synthesis, here is the lactate, then the glycerol, and after this glycerol I have added the lactate synthesis. So when you will read the topic of the slide of lactate synthesis, then you will see this table, and when you will see the glycerol synthesis, then you will see this table. Then how lactate is converted into the glycerol, only when you will learn the tables, you will learn most of the lecture. So lactate, how lactate is converted? Lactate is converted into the glucose. How it is converted? Lactate is converted by only one step is different here. The one step that is different is lactate is converted to pyruvate, then pyruvate is converted to, all the steps are the same, the rest of the steps are same. Pyruvate is converted to oxaloacetate, and then oxalic acetate is converted into the phosphoenol pyruvate, and the whole process is reversed. Then this is the conversion of lactate to the glucose, in which only first step is different, in which NADPH is utilized and lactate dehydrogenase enzyme is used. So lactate is converted into the glucose in this manner. I have added it in the slide. In the written form, lactate is converted to pyruvate with the help of reversible reaction. The enzyme required for this step is lactate dehydrogenase. In this step, NAD is converted to NADI. This pyruvate is converted into the, then pyruvate is converted to oxaloacetate, and oxaloacetate into the PEP, that is a phosphoenol pyruvate, and the remaining steps are repeated. Next, how glycerol is converted into the into the glucose? We can see that glycerol is converted into the glycerol 3-phosphate. Glycerol 3-phosphate is converted into the dihydroxyacetone phosphate. Basically, this is the enzyme, glycerol 3-phosphate dehydrogenase enzyme. So when you will draw in your exams, you will draw like this: glycerol is converted into the glycerol 3-phosphate, and glycerol 3-phosphate is converted into the dihydroxyacetone phosphate, and then dihydroxyacetone phosphate, which is produced during the process of glycolysis, then the whole glycolytic pathway is reversed here. So this is the enzyme here. See, you will write like this, that glycerol is converted to glycerol 3-phosphate, glycerol 3-phosphate to dihydroxyacetone phosphate, and in this step the glycerol 3-phosphate dehydrogenase enzyme is used. Next, you can see that how propionyl CoA is converted into the for the production of the glucose. Here propionyl CoA is converted into the propionyl CoA is converted into the glucose in such a manner that propionyl CoA is converted into the methyl malonyl CoA. Then methyl malonyl CoA is converted into the succinyl CoA, and we know that the succinyl CoA is an intermediate in the TCA cycle. So we know that from succinyl CoA oxaloacetate is formed, and from oxaloacetate phosphoenol pyruvate is formed, and phosphoenol pyruvate enters the glycolytic pathway, and the whole glycolytic pathway is reversed. And remember one thing, when you will write in your exams, then you will write the whole glycolytic pathway here up to the phosphoenol pyruvate. For the conversion of propionyl CoA to the methyl malonyl CoA, there is an enzyme known as propionyl CoA carboxylase, and as I have told you right sometimes earlier, that carboxylases requires A, B, C, that means ATP, biotin and carbon dioxide. From methyl malonyl CoA, it is converted into the succinyl CoA, and here the enzyme is mutase, and vitamin B12 is also required here. So the rest of the steps are the same as we have discussed earlier, that succinyl CoA is converted into the oxaloacetate as it is converted in the TCA cycle, and then oxaloacetate is converted into the phosphoenol pyruvate with the help of enzyme as I have discussed it. So that was the conversion of this propionyl CoA, glycerol and lactate into the glucose. This is the slide of how glycerol is converted, and I have added slide here. I have discussed it earlier. So when you will see the table, then you can read this slide of glycerol and can remember it. This is the slide of lactate. Some amino acids are also used for the synthesis of glucose molecules. How these are used? Alanine, serine, glycine, cysteine. These are the 4 amino acids. Basically, these amino acids required for the production of pyruvate, and then pyruvate is converted into the glucose as I have told you earlier. Similarly, okay, there is one thing here, that alanine, this enzyme is the most important enzyme, and leucine is the only amino acid which is the ketogenic amino acid. Remaining all amino acids are the glucogenic amino acids. Next thing is succinyl CoA. How succinyl CoA? You can see valine, methionine, isoleucine, threonine. These amino acids are converted into succinyl CoA, and I have told you earlier that succinyl CoA is how succinyl CoA is converted into the glucose. Next, alpha ketoglutarate. We know that alpha ketoglutarate is also an intermediate of TCA cycle. So glutamine, proline, histidine, these 4 amino acids are responsible for the conversion into the alpha ketoglutarate, and the alpha ketoglutarate then in the TCA cycle is converted into the oxaloacetate, and then oxaloacetate enters the glycolytic pathway. Similarly, fumarate is formed by the Triosine is responsible for the, and phenylalanine are responsible for the formation of fumarate, and aspartate and asparagine are responsible for the formation of oxaloacetate. When fumarate is formed, it is also an intermediate of TCA cycle. It enters the TCA cycle and then ultimately converted into the oxaloacetate, and then oxaloacetate is converted into the glucose. And when aspartate and asparagine is converted into the oxaloacetate, then we know that oxaloacetate is converted into the phosphoenol pyruvate, and that is responsible for entering into the glycolytic pathway. So these were the steps, for the 4 steps mainly that were responsible for the production of glucose from non-glucose sources. Now we will discuss about the very important short question related to the gluconeogenesis, that how gluconeogenesis is related. There is the induction and repression. Insulin inhibits pyruvate carboxylate. So inhibits gluconeogenesis. Similarly, glucagon induces this process. So glucagon is responsible for inducing this process. Some covalent modification occurs. Protein kinase important reaction occurs. Cyclic AMP, that is the cyclic adenosine monophosphate level is increased, and that increases the cyclic AMP dependent protein kinase enzyme. Then phosphorylate these enzymes in gluconeogenesis, and then the process is regulated. Similarly, allosteric regulation is also there. Acetyl CoA is activated of carboxylase, so it activates the gluconeogenesis. Similarly, fructose 2,6 biphosphatase is a phosphate is inhibitor of pyruvate carboxylase, so it inhibits this process, and in this way gluconeogenesis is regulated. So regulation involves the enhancement and the reduction, that how this process is induced and how this process is reduced. So this is the short question, and you can if you will only write these slides that I have added here, then you will get the maximum marks, inshallah. That was all about the gluconeogenesis, and if you have any queries related to this topic, you can ask me in the comment section. Thank you very much for watching.